Usually I have to spend 20 or 30 slides when I give a talk, just convincing everybody that the future of medicine is here. So it's just awesome to come here and actually cut those first 30 slides out and get right to the meat of it. So I'm going to talk to you about precision medicine, like several folks here that have given a talk on precision medicine and this idea of precision medicine, but I also want to talk about precision wellness, which I think is really an evolution or a next step of precision medicine. I'll get into that in some detail here. So one thing that really keeps me up at night as we're racing towards the future of medicine is this worry or this concern that are we plotting a course to the future of medicine with the wrong map? And what I mean by that is we've been making great strides leveraging digital health, using things like genomics to really focus and better understand disease. And this is a lot of work that I do. How do we apply metabolomics, proteomics, genomics to better understand disease? But all this work is really focused around the idea of a disease and the diseases in the way that we've defined them, right? Which are largely symptoms and anatomy based. You can go back to these old drawings from the 1400s on diseases and anatomy, and what you'll find if you look at all these really interesting drawings is that they're very anatomical focused. Disease is all about symptoms and anatomy and locations of the body. And if you go into a hospital today, what are you going to see? You're going to see signs that say urology, cardiology, neurology. Everything's still symptoms and anatomy oriented, right? And our diseases are still oriented around this taxonomy of disease. But what we know from genomic studies is that the symptoms and anatomy orientation of disease around which all of our efforts are being focused is really incorrect. So this is work I actually did with Atul Butte, who you heard speak last night as the keynote speaker, was leveraging all the genomics data we had and looking at diseases from a genomic perspective in terms of pathways and genetics and things that are being disrupted at the molecular level, and then comparing across diseases at the molecular level and seeing how does the genomic data-driven taxonomy of disease organize, and how does that compare with our symptoms and anatomy-based taxonomy of disease? And what you'll see is, for example, if you look on the right there, Alzheimer's disease is connected to actinic keratosis. Actinic keratosis is a skin disease. So what that line connecting those two things is saying is that at the genomic level, in terms of the molecules, the genes, pathways that are being disrupted, if you look at these two diseases, they share a huge number of commonalities in terms of what's being disrupted or broken. It seems very bizarre, but actually, if you know your developmental biology, the brain and the skin actually come from a common sort of structure in development called the neuroectoderm. They have a common sort of stem cell lineage, and they go their separate ways and become the skin and the brain. So maybe it's not so crazy that these things are breaking down at the molecular level in different tissue contexts. So it looks like different diseases, but at the molecular level, it's sort of the same parts that are broken in a different context. And there's a group in Mexico, actually, several months ago that started looking at the skin of Alzheimer's disease patients. And they claim in that study that they can see they could see tau tangles, which is a form or a sign of Alzheimer's pathology in the skin of Alzheimer's patients at around the same time that their brains are developing Alzheimer's disease. So not so crazy that the skin and brain are connected. But really what the opportunity is now is that we can bring data. So now we ourselves and others have brought lots of genomic data to really challenge this idea that this sort of taxonomy or landscape of diseases is really what we thought it to be. And there's really no such thing as a simple disease. When we think about genetics and disease, one thing that people often do, and geneticists I think are guilty of this, is organizing diseases into simple genetic diseases and complex genetic diseases. A simple genetic disease like cystic fibrosis, what this is showing here on the left is this picture is all the systemic manifestations of cystic fibrosis. Cystic fibrosis is a disease where we have one gene, a mutation in one gene is the single causal genetic driver of this disease. We've known this for almost 20 years. So look at all the complexity across all these other organ systems, interactions with other genes that can emerge from this single mutation. By the way, we've known this gene for almost 20 years. We still can't cure the disease. And now we have diseases like Crohn's and diabetes, where we have hundreds of genes underlying this disease. Woody Guthrie had Huntington's disease, the same thing, right? So even though it's a single genetic cause, unbelievable complexity. And humans are complex adaptive systems. So we really have to appreciate not only humans, but human populations as complex adaptive systems. Complex adaptive systems have characteristics. They evolve over time. There's interactions. The whole's always greater than the sum of the parts, and you cannot understand the whole system by looking at its parts. This is a critical factor, and I always like to point out who said this is an investment banker. So for as much as biologists and clinicians will make fun of bankers, they're really on the forefront of embracing and appreciating complexity and complex systems. So really, this is how we approach everything in medicine where I work at Mount Sinai, is sort of a pro-complexity stance. Is that if you just think of one cell in the body, just a single cell, You have billions of bases of DNA. You have those expressing RNAs. You have networks of RNAs. They make proteins and metabolites. There's thousands of millions of interactions between them, among them, protein binding to DNA. You have a whole universe of constellations of interactions happening in just a single cell in the body. And they organize into tissues and communication across tissues and communication across organs. And of course, the environment interacts with that from all sides. So to really organize these diseases around symptoms and anatomy, when we understand that everything is connected, is a bit silly. And we've made great strides in systems biology, where we can look at, for example, in a population, and maybe that red thing is a base of DNA we've measured on a population, and the green thing is a metabolite, and the blue thing is a voxel in a brain imaging scan. And we have systems biology methods that let us really understand the molecular complexity of disease and build these biological networks and really model through graphical modeling algorithms, et cetera, how at the molecular level all these things interact and are put together. But I would argue that now that we've made great strides in systems biology, we need to keep pushing this idea of embracing complexity and modeling complexity up beyond just cells and individuals, up to populations. And this is really the idea of personalized or precision medicine. So really understand now things at the population level. We have a great resource at Mount Sinai called the Mount Sinai BioMe Biobank. We have about 30,000 individuals who have consented, and it's a very diverse group of individuals ethnically, have consented to give their DNA, and we can actually sequence their DNA, which we've done, and have genotyped them all and have tied to their electronic medical records, similar to efforts that Dr. Green was talking about. So when I initially got access to this resource, I saw it as a great opportunity to explore this idea I think that many people in this room are interested in, of a learning healthcare system. What drives me absolutely bonkers is if I go to a hospital, they'll say, okay, Joel, you're a 39-year-old Caucasian male with a family history of X. Now let's start treating you based on guidelines of what we know about people like you, right? Things that have been published in New England Journal of Medicine or by the various societies. But what they don't do is say, wow, we've seen millions and millions of patients at Mount Sinai. Mount Sinai is the biggest healthcare provider in the state of New York. We see millions of patients a year. What patients look like you and whatever happened to them. We never do that in a data-driven way, right? But so this is a first attempt here at trying to enable this idea of a learning healthcare system through the lens of precision medicine. And what you're looking at here is each dot is not a gene, like in a genetic network. Each dot is a patient. So what I did is I took all the patients and I took all the clinical data I had on them. So things like complete blood counts, metabolic panels, everything that was a sort of objective clinical measure, height, weight, ethnicity, et cetera. And then I created a huge repertoire, a vector of information, and I said across these hundreds and hundreds of clinical features, which patient looks like this patient, looks like this patient, looks like this patient, looks like this patient, right? And I built a patient-patient similarity network. It's almost like a social network where you're connected to someone because they have the same friends or same interest in movies and books, except in this case, you're connected to someone because you have the same blood test results. And now I ignored whatever the diagnosis codes were applied to these patients. I said, I don't care what the doctors think, what disease the doctors think they have. All I wanted to focus on was I have these raw measures on these individuals, these blood test results, and I wanted to let the data tell us how the patients are organized without any preconceived notion about disease. So the X and Y don't mean anything here. The only thing that's important is the topology. I have no idea why it looks like South America or a big fish. That's just the way the algorithm works. It's not geographic. But then what I did here, well, broken it into two groups. So now if you think of yourself almost like in a Google Maps context, right? If you're a new patient at Mount Sinai, the first question is, where's my little blue dot? Where's my little blue blinking dot on this, right? Because these are all these other patients. I want to know. I can tell you, you want it to be over in that blue zone. You don't want to be over on that other island. I have many bad names for that island. The nicest thing I can call it is cardiometabolic island. It's a bad place to be. And even if you go towards the shore there, it's a bad thing. So, and you want to think, where's my little blue dot moving? You know, and it doesn't mean anything, right, amongst all these patients. So what I did here, the coloring is highlighting the patients with type 2 diabetes. Now these are patients that have the traditional narrow definition of what is type 2 diabetes, HbA1c results of X, glucose results of X, and we highlighted where are these traditionally defined type 2 diabetes patients, and the colors actually represent hotspots of type 2 diabetic patients. In the red zones, it means that there's great concentrations of type 2 diabetes patients in our current definition, more than you'd expect by random chance. And because I have genetics on these individuals, which was kept separate from this clustering, the genetics were not used to cluster these patients, I could grab that group on the left there, that group on the right there, and say, let me do a genetic association on this sort of data-driven emergent disease population from the population, rather than using these narrow phenotypes. But the big idea behind this is this. Now I've subtracted out all the patients that don't have our traditional definition of diabetes, and I'm just focusing on what are the traditional diabetic patients. And again, I'm letting the data organize these clusters, and they consistently break into 3 different clusters. So even though these are thought of as one disease in the clinic, they consistently break into 3 different groups. And now it's being colored by male and female, just to show you that actually male, gender, ethnicity, race are really not driving, in this particular case, these clusters. So why should I care about these clusters? Well, we have electronic medical record data on these patients, so we could go back and compare. Are there any clinical differences among these groups that we can see in the electronic medical record that are of clinical importance? So for example, the group at the top there, if you control comparing to other groups by age, disease onset, et cetera, they have much greater relative risk of cardiovascular complications. So all type 2 diabetics have risk of cardiovascular complications, microvascular like retinopathies, nephropathies, et cetera. But these have, even relative to other type 2 diabetics, greater risk. So that's interesting. And the bottom left group there is actually a very high-risk cancer group. So there's an interesting axis that connects diabetes and cancer. Diabetes increases your risk of cancer. The diabetes drug metformin actually decreases your risk of many cancers across the board. Nobody really knows why. Some theories about Warburg effect, et cetera. But so this bottom left group has much higher risk, even though all diabetics have increased risk of cancer, this group has much higher risk than even relative to other diabetics. So this is the idea here. It has type 2 and type 3 and type 4 diabetes been right under our noses the whole time at Mount Sinai, but we never thought to go back and look at our own data and learn that these patients exist. We just took for granted these standard definitions of these diseases. So remember, we had genetics on these individuals. So then what I could do is I could do a genetic association on these data-driven disease groups. And I find genetic factors that cluster or that are found uniquely in one group versus another. So we found many genetic factors that were unique to each group, genetic variants. And when we looked at the biological significance of these genetic factors that were unique to each group, what got us really excited about this, and why we're lucky enough to have the director of the NIH blog about this paper today, was that the genetic factors seem to corroborate the clinical differences. So I'll get specific in a second here. So remember, the genetics were kept separate out from this clustering. They did not determine the clustering. So, but what we found is, for example, that top group that had increased risk of cardiovascular complications, the genetic factors that were unique to that group were very significantly enriched in things like vascular endothelial signaling pathways. So this is sort of the lining of the vasculature, right? So it's really showed that these clinical differences that we observed among these groups are probably not just random correlations, but there's actually some underlying biology that probably drives these differences. So I think, as this develops and in the future, as you're a newly diagnosed type 2 diabetic person, I hope what we're able to do is use these clinical and genetic factors and say, oh, you're a type 10 diabetic. And for type 10 diabetics, we should really monitor cancer more. We should use this drug or instead of that drug, et cetera. So this is really where this is going. And we've done this now with other diseases like cancer and multiple myeloma. I'm going to go through this quickly for the sake of time, where we're also looking at cancer patients in this way and making drug predictions now. So we're actually clustering the patients and picking drugs in terms of where they fall in these phenotype clusters and not based on guidelines. This is a real patient at Mount Sinai. I can go through quickly, where we repurposed a drug from another cancer. But this is her disease markers. This is a free lambda chain skyrocketing. We made a prediction based on the cluster she lived in of a certain drug. We gave her this drug that would not have been used for multiple myeloma. She was checking into hospice at the time, and then based on this drug that was chosen based on where she clustered in these types of groups, she went to Hawaii instead a couple weeks later. But I'm actually shocked that we got anything out of this data, because electronic medical record data is such garbage. It's so sparse. It's so biased towards billing. I mean, the 364 days of the year that you're doing other things besides going in the hospital, it probably has a huge impact on your health. I'm actually shocked, to be honest, that we got anything useful. So what we're really appreciating now going forward is this need to really fill in that gap, right? So we're really good with genotype and molecular data. We have a paucity of phenotypic data. So I always like to create this contrast in people's mind of a Formula One race car and all the data feeds we have, the custom iPad apps, the Oracle boat that just won the America's Cup race not that long ago, all these sensors, all this information, custom iPad apps. This is what happens when you're born in a hospital today in the United States. You get your little APGAR kind of score components. You heel prick, maybe we'll look at PKU. Hopefully look at the whole genome if Dr. Green is successful. And you get your birth certificate and the slap on the rear end and say, come back and see us when you're sick. I'm going to go, for the sake of time, fly through this, but everyone in this group appreciates that we have great opportunities to fill in this phenotypic space with consumer-centered technologies. Even diapers are going to be smart. Consumer healthcare is going to be a big driver of data-driven medicine and enable us to do things that would be difficult to do inside institutions. And what I really want to focus on now, going forward, is definitely precision medicine, but this idea of precision wellness, to collect long dynamic data to enable this precision wellness. So we're really focused on this nexus of digital health, data science, and molecular profiling in clinical medicine that is making big strides in focusing on diseases like asthma, cancer, et cetera. But I think there's a really huge opportunity to take these same powerful tools and the same hard scientific approach to focus on a normal, healthy person and wellness. We have no idea what a normal, healthy person is. The dynamics and variability and complexity we would see in normal, healthy people, I think, is tremendous, but it's completely blind to us now. And how do we better understand these transitions to disease rather than focusing on people who have already fallen off the cliff? So remember this map I showed you? This is cool, and you can sort of think of like this is like ways for health, and we want to find like where we're navigating on this map and how we're moving across it. But this is all sick people, right? So this map, while it is cool, is very similar to the map I showed you in the beginning of that sort of old map that Columbus might have used that was Eurocentric, Asia-centric, that didn't even have America on the map, right? So this map is just as rudimentary as that because it's not been filled with all the variability and the landscape of normal, healthy people. And I think we really need to focus on increasing the well span. So if you look at, this is people with the number of chronic diseases by age and percentage of the population, and all of our NIH research and all of our pharma development and et cetera is all focused on the upper right, and it should be because people have those diseases. But if you're lucky to be born on that trajectory or that trajectory, your life is very different. So instead of really focusing all of our resources on the upper right-hand quadrant, why can't we focus a lot of our resources on increasing that green space, right? That seems to me to be a very rational thing to do. And if we think about networks, think about these complex networks of wellness, right? So we have things like genetics, genomics, immune system, sleep, cognition, and what I was showing you there is we have tons and tons and tons of quantitative data on genetics and genomics. We have almost no quantitative data on any of those other dimensions of wellness. Think of your immune system. Your immune system protects you from pathogens, from the environment. It's scouring for cancer cells in your body. It's doing all these different things. When's the last time you got a checkup for your immune system? Do we even have tools to do a checkup on your immune system? If we know it's one of the most important things that's influenced by sleep and metabolism and mood, and that your immune system affects cognition, et cetera. So what we're trying to really enable, like I mentioned before, is this more real-time monitoring of health and wellness, and not wait till you've fallen off into these cliffs of disease, but how do we keep you on the optimal healthy path, right? How do we help you navigate this topology in a data-driven way and modify your personal internal networks, your environmental networks, your social networks to keep you on the optimal healthy path in a hard, data-driven, scientific way. And I just go to the end here, because there's apps here, people focusing on this. There's a lot of great work focusing on apps to make you run more, to exercise more. That's all great. But I mean, this sucks, right? I mean, it's not the future I wanted. The future I want in terms of wellness is I want a probiotic ketchup that I put on my cheeseburger that excretes PCSK9 inhibitors so I can eat cheeseburgers all damn day long and not.
I want the card.
Right? Right, so that's what we should really be aiming for, right? So we're running a study here to actually build this map with data. And really to build out this wellness map or this whole map of human wellness and disease and really help understand for individuals where do you live on this map, where are you on the well map, and how through algorithms do we help you navigate this map in a smarter way. So thank you.
Thank you. Yeah. Thank you very much.
There's my bold prediction.
Thank you. Thank you. Joel Dudley. Very good.